We continue our discussion on enzymes. In this particular lecture, what we are going to talk about is something related to the enzyme substrate interactions, discussions on how they have their specificity, stereospecificity, and factors that would influence enzymatic activity and how they may be regulated. Catalytic activity and active sites of enzymes in terms of the specificity or what we have in terms of geometric complementarity is something extremely important for the enzyme substrate complex to form, which is a prerequisite for any enzymatic activity. Proteins as enzymes. All enzymes are proteins except a small group of catalytic RNA molecules known as ribozymes, which we will discuss briefly, and we will look at their mechanism when we look at enzyme mechanisms in general. The catalytic activity of enzymes depends upon the integrity of the native protein conformation, and with the dissociation or denaturation of the enzyme, the catalytic activity is lost. We realize this because there is a geometric component to the specific enzyme that holds the specific residues that are involved in the mechanism in place. Breakdown of the structure, therefore, would result in a loss of the catalytic activity. The enzymes, like proteins, have large molecular weights, but their specific zone of action is limited to the active site, which is why there is a lot of research in the area of looking at mimicking enzymes where only the active site residues are taken into consideration to bring about a catalytic activity in mimicking the enzymatic function. Enzymes differ from ordinary chemical catalysts in several important aspects. They have higher reaction rates, they have milder reaction conditions, they have greater reaction specificity, and there is a capacity for control. If we look at the generic working of the enzyme, we will see that there is, as we look at here, a specific methodology where we understand that the enzyme and the substrate together is going to form the enzyme substrate complex. This may form an enzyme product complex where the catalytic mechanism of the enzyme occurs in this step, which then breaks down to the E plus P. Now, all of these do not involve exact pre-equilibrium. This is a pre-equilibrium that occurs, but all of these may or may not involve a pre-equilibrium step. This, therefore, has the structure where we can look at the enzyme. We have the substrate, we have a fit, and then we have the breakdown of the enzyme to release the product. What is important here is in the catalytic activity of the enzyme is the enzyme itself is restored to its original conformation in terms of the active site residues in their specific geometry, because the enzyme has to be ready to bind another substrate. This is the beauty of enzyme catalysis. So if we look at enzyme substrate interactions, we see we have an active site and we know that this active site is going to bind our substrate. Subsequently, we would have product formation. We could have various types of product formation. We could have one, we could have two, depending on the type of mechanism that occurs. If we look at enzymes that change their conformation due to the presence of the substrate, we form what is called an induced fit mechanism, where in this case the enzyme accommodates to take in the substrate molecule and we again have product formation. The binding of a substrate to an enzyme at the active site is therefore an extremely important component. The molecular recognition, as we spoke about in protein ligand interactions, where we realize that the binding of the ligand, in this case the substrate, to the enzyme is an important factor in the recognition, and in this case, a particular reaction to take place to form the product. So when we look at the substrate bound to the enzyme, in this case chymotrypsin, we realize that there is the specific active site residues that are involved, the important residues that are involved in the recognition of the substrate to the enzyme. So we see that even though this protein is very large in its size, there is a specific region that is the active site that is required for the recognition of the substrate. The substrate specificity, therefore, is important because there is this important role of recognition. So we have the substrate, we have the product form, and we have the enzyme as the catalyst that works here. So the non-covalent forces through which the substrates and other molecules bind to the enzymes are maximized to bind the substrates with considerable specificity. So we have van der Waals forces, we have electrostatic forces, the ionic interactions, and hydrogen bonding. And we realize that since we have the enzyme substrate complex and this dissociates to give the product, we cannot have the ligand binding, in this case the substrate binding, to be too strong to the enzyme. However, when we are designing an inhibitor, we would like the binding to be strong. This is something we will discuss when we look at the specific enzyme kinetics and enzyme mechanisms. And so these are the types of interactions that is possible, the non-covalent forces that would result in specific substrate recognition of the enzyme. There are some enzymes that are involved also in covalent linkages, but during the catalytic mechanism, they revert back to their original enzymatic structure. So we look at our substrate and we have our enzyme. We realize that it is a complex that illustrates not only physical complementarity, chemical complementarity, geometric complementarity in bringing the formation of the enzyme substrate complex. So if we have hydrophobic groups, we can have hydrogen bond formation, and we have stereospecificity, a very important factor in the chirality that results in these molecules. Now, enzymes are known to be stereospecific. Now, this stereospecificity arises because the enzymes, by virtue of their inherent chirality, form asymmetric active sites. So what we have is we can have a situation where we would have the yeast alcohol dehydrogenase that catalyzes the interconversion of ethanol to acetaldehyde. Now, if we look at this particular interaction, we have to understand what we mean by prochirality. In prochirality, what we have now is we have two substituents R1 and R2 to the carbon, and we have two hydrogens attached. Now, this is called a prochiral carbon because this replacement of one hydrogen is going to result in a chiral carbon. So now if we look at these prochiral hydrogens, we realize that the loss of or the change of one of these hydrogens is going to result in a chiral carbon because now we have the four substituents that are different. So this occurrence from achiral to chiral in the substituent changes is important, where we can form the pro-R and the pro-S. What do we mean by these? So now if we have a representative structure that tells us this is our CH3, CH2OH, our ethanol. What is going to happen here is if we change H to D, then we will have this specific conformation. We now have a chiral carbon. We have an R configuration. This means that since we change this particular hydrogen to the deuterium, the change of H to D has resulted in the R configuration. So this is called the pro-R hydrogen. Similarly, if we have the change of the other hydrogen to deuterium, we result in an S configuration. This S configuration for the carbon means that the specific hydrogen is the pro-S hydrogen. Now, if we have two identical substituents that are attached to an sp3 hybridized carbon atom, the descriptors pro-R and pro-S are used to distinguish between these two. Now, promoting the pro-R substituent to higher priority than the other identical substituent gives us R chirality as we saw, and similarly we can do it for the S one. Now, we can have two phases of the molecule. We can have the re phase or the C phase. So the trigonal planar sp3 hybridized atom can now be converted to a chiral center when we have the substituents attached from either phases. So if we look at how they are labeled, when we look at the face, the substituents at the trigonal atom are arranged in decreasing priority order, that's 1 to 2 to 3, in a clockwise manner. Then we have this phase, and if they are arranged in an anti-clockwise manner, then we would have the C phase. So if we look at the specific reaction now, where we have the conformation, the enzymatic activity occur in yeast alcohol dehydrogenase, there is a way in which we can form from the substituent ethanol to the substituent acetaldehyde, giving us our overall reaction. And from this, we understand that the pro-R hydrogen is removed, and this, if the particular enzymatic reaction is carried out with deuterated ethanol, we see that the product NADH is deuterated. So this gives us an idea about the enzyme activity, about the enzymatic mechanism. So there is a differentiation by the enzymes, and the specific attachment of a prochiral center to the enzyme binding site permits the enzyme to differentiate between prochiral groups. So we understand the specificity of the enzyme and how important it is to get the specific chirality or the specific configuration of the molecule of interest. So when we look at the side additions also from the two different sides, then in this case, we get identical products where we have a pro-R and a pro-S. What is known is that most reactions with an equilibrium constant for reduction that is greater than 10 to the minus 12 use the pro-R hydrogen, and those with reactions with less than 10 to the minus 10, they use the pro-S hydrogen. So the specific residues that we have help maintain the stereospecificity. For example, the liver alcohol dehydrogenase makes a mistake 1 in 7 billion turnovers. So the efficiency is remarkable. Now, if there is a mutation, which means that one amino acid has been changed to another amino acid, in this case, a hydrophobic leucine has been replaced by a hydrophobic alanine, it increases the mistake rate. As a result, there is an 8,000-fold increase in the mistake rate. This suggests that the stereospecificity is helped by the specific amino acid side chains present. So, apart from the chemical specificity, we understand the importance of the geometric specificity. Here, there is a selectivity about the identities of the chemical groups. But enzymes are not always molecule specific. There is a small related compounds that will undergo binding or catalysis. Say we are looking at chymotrypsin. We have the peptide bond and we have the cleavage of the peptide bond. Again, we could have the ester and the cleavage of the ester. But we have reaction specificity. So if we are looking at an oxidation reduction redox type, we would have, say, ethanol go to acetaldehyde. If we are looking at a group transfer type, we would have oxoglutarate in this case forming L-alanine would form the, so we have the L-alanine L-glutamate where we have a group transfer. Now all of these reactions, as we looked at in the previous lecture, are important enzymatic reactions belonging to the specific classes of enzymes that we spoke about. So the specific classes have reaction specificity. The third type was hydrolysis, brought about by hydrolases. So this is a specific hydrolysis reaction. Similarly, we can have ligation, we can have isomerization. And so in this case, we would have, remember we used ATP for the specific energy requirement for the formation of this bond. So the linking is going to result in energy from the cleavage of the ATP to form ADP and PI. Now, enzymes, the important part is they affect reaction rates, not the equilibria, which is something that we will understand greater when we go to do the kinetics. So to understand catalysis, we have to realize that there is an important distinction between the reaction equilibria and the reaction rates, something that we also discussed in our discussion for protein ligand binding, where we have a reaction coordinate diagram that speaks of the free energy change. So we have the ground state for the enzyme, we have the ground state for the product that is more stable than the enzyme. Then we have a free energy change associated with the process. What the reaction coordinate diagram, when we have enzyme catalyzed and enzyme uncatalyzed reactions, tells us that we have variations because of the catalysis brought about the enzyme that makes the reaction easier to occur. We will speak about this in further detail when we study enzyme kinetics. We have in this case the reaction intermediates, the enzyme substrate complex. There is also possibility of enzyme inactivation. This is going to be important in inhibitor design. When we want to have an idea of the active site, we know the substrate binds to the active site. But if we want to inactivate our enzyme, we have to design an inhibitor. The inhibitor design can be a substrate analog. But in this case, we would like the enzyme inhibitor complex that is also occurring because of molecular recognition to be a tight complex. So we can result in a protein shape difference, an enzymatic active site geometrical loss, the scaffold being lost because of the external stress on the protein. For example, by applying heat or changing the pH, what will happen, as we studied earlier, is we will have a denatured protein, and this denatured protein cannot carry out its cellular function. For example, The most common example is where we have irreversible egg protein denaturation because caused by high temperature. So factors that influence the enzyme activity are similar to those that would be involved in a protein. Temperature and pH, any change would result in a loss of structure, a loss of function, in this case a loss of enzymatic activity, and other factors, coenzymes and inhibitors. So we would have an optimum temperature for enzyme activity, which we realize is around the functioning around our body temperature, around 37 degrees centigrade. Similarly, we would have an optimum pH for most enzymes around 7 to 8, 7.4 being the physiological pH. Another very important enzymatic class that has gained importance over the past years are ribozymes. These are catalytically active RNA molecules or RNA protein complexes. And the term ribozyme refers to the enzymatic activity and ribonucleic acid nature at the same time. There are small ribozymes that are hairpin ribozymes, hammerhead ribozymes, and the hepatitis delta virus type. There are large ones also. The large ribozymes are the ribosome, spliceosome, and RNase P. Now, these are also very specific in their activity, in the sense that if we look at the structures of these are RNA structures, a lead zyme, a hammerhead ribozyme, and a twister ribozyme. The beauty of these is that these RNA molecules have the ability to catalyze specific biochemical reactions, just like enzymes that are proteins. We have RNA splicing in gene expression, viral replication, and the transfer RNA biosynthesis. So the most common activities of natural ribozymes are the cleavage or ligation of RNA and DNA and peptide bond formation. So these are all catalytic reactions, and in this case, the catalytic catalysis is brought about by ribozymes. For example, if we look at RNase P, it's a site-specific endoribonuclease which cleaves RNA. So it has a specific structure, a specific metal ion involved, and this ribonucleoprotein, RNA and a protein containing complex, and the catalytic component in this case is RNA. We can have cleavage of typical pre-tRNA by ribonuclease A, and these are the specific substrates. It is the RNase P substrate, and there is a cleavage site that belongs to the recognition here are the RNA bases. Similarly, there are other types where we have the RNase P substrate and we have the cleavage site for an RNase MRP substrate. So these are specific reactions involving RNA and the catalysis brought about by these. Like many protein enzymes, metal binding is also critical to the function of many of these ribozymes, and they can also catalyze the formation of the peptide bond between adjacent amino acids by lowering the activation entropy, which is what happens in ribosomes. And within the ribosome, ribozymes function as a part of the large subunit ribosomal RNA to link amino acids during protein synthesis. So, if we look at the ribosome, that is this wonderful biological machine that utilizes a ribozyme to translate RNA into proteins. Here, the central catalytic activity of the ribosome, that is the peptide bond formation, is catalyzed by the RNA component. So as we see the increase in the formation or the linking of the formation to form the peptide, we realize the importance of the ribosome in the formation of our proteins. So we have a regulation of enzymatic activity, There are two general ways to control enzymatic activity: controlling the amount or the availability of the enzyme, control or regulate the enzyme's catalytic activity in several ways, and each topic can be divided into several subcategories, which we will visit later on. So the regulations can be done by these small molecules that can act as competitors, allosteric molecules that would act at sites away from the active site of the protein, but nevertheless affect the enzyme activity in terms of enzyme inhibition. Phosphorylation, where we could change the enzyme activity; degradation, denaturation of the enzyme that will result in a loss of the scaffold that holds the active site residues in position to recognize the substrate. That is also a very important factor. And the synthesis can also be changed. Now, the geometric specificity and the chemical specificity are therefore extremely important for an enzyme to recognize a substrate, to form or to undergo its catalytic action. The enzyme mechanisms, which we will be visiting in the subsequent two lectures, that will tell us about how specific the substrate is for a particular enzyme. These are the references. Thank you.